Okay, in Chapter 4 we're going to have an introduction to cell structures. I'm not going to explain every cell structure in one chapter, but we're going to lay out some of the basics, and that's what you need to learn, starting with we're going to look at a little bit of structures that are in all cell types and then some structures that are in certain cell types. So primarily the division of cell types that we will look at will be bacterial cells, which on this screen is shown on the far right. So this is an image of bacterial cells here on the far right, letter C. In the middle, it's a kind of a pretty standard picture of some plant cells. And then on the far left, some animal tissue. So the way cells look can be quite different. And one thing to pay attention to as well is the scale. So on A, the little scale legend has a measurement of 10 micrometers. That's the Greek letter mu. It looks kind of like a lowercase u. So 10 micrometers. In the middle, about the same measurement, is 50 micrometers. And so plant cells are typically bigger than animal cells. And then on the far right, on C, this is 500 nanometers, is the same little length. Now a nanometer is 1/1000th of a micrometer. So a half of a micrometer. And so bacterial cells are actually significantly smaller, and so much so that you would never mistake a bacterial cell for a eukaryotic cell. And of course, plants and animals are both eukaryotic. So how do we make pictures like the ones I just showed you? We use microscopes, and a picture that's made with a microscope, an image, a photograph made through a microscope, is called a micrograph. So sometimes you will see that word, a micrograph. Micrograph. So if somebody refers to a micrograph, it's a photograph that was taken looking through a microscope. So in any case, you have two types of microscopes. The ones we would have used in lab would be the light microscope, sometimes called the compound light microscope. You have objective lenses of different magnifications on the nose piece. Those are here. And then you look through the eyepiece here, or the ocular. And the eyepiece has it depends on the scope. The ones we use have a magnification of 10 on the eyepiece, and then on the objective lenses you would have typically 10, could be 4, sometimes 4, 10, 40, and 100. Those would be pretty common. But different microscopes can have some different lenses. In any case, it's called a compound light microscope. Light because there's a light bulb at the bottom, so the light shines up through, there's a hole in the stage, which is the flat black surface, and then the light goes up through the objective, and then it goes through the ocular lens to your eye. So it passes through what we call two image-forming lenses, the objective and the ocular, before it gets to your eye. And it also passes through the object that's on the slide. The slide is on the stage. Now, the electron microscope, this is a picture of an electron microscope, and they're quite large comparatively. It's much more expensive. A light microscope can cost anywhere, $5,000, something like that. I think the ones we have in lab are for $4,000 or $5,000 a piece. An electron microscope is hundreds of thousands of dollars. So this is not something that you have typically in a student lab. In fact, we do have one on campus at Richland, but if you go to like UT Southwestern, they have them, but not very many because it's such an expensive tool. You have to prepare the slides much differently, and what happens in an electron microscope is a beam of electrons, instead of a beam of light, a beam of electrons is passed through the slide, and the electrons are deflected to create the image. But your eye doesn't ever look directly at the beam of electrons. Your eye looks at a screen that shows the image. And there's two kinds of electron microscopes. One's called scanning and one's called transmission. A scanning electron microscope, SEM, scanning will show you a three-dimensional image of the surface of an object. So on that last slide, like this one here, this probably was done with a scanning electron microscope, and then you only see kind of the surfaces of the objects. And the other thing is, it always gives you a black and white image on an electron microscope, but they typically will colorize the image using software. A transmission electron microscope gives you a 2D image. It almost looks like a slice through the object, so a two-dimensional image. Transmission electron microscope. So scanning gives you a three-dimensional image of the surface. Transmission gives you a two-dimensional image, like a slice through the object. Kind of like when you get an MRI, they don't actually slice you, but the radiation passes through and gives an image of a slice of you. So, and they both the electron micrograph types will be black and white, and then they colorize them. So when you see SEM or TEM in your book, when you're looking at an image, that tells you how the image was created, what kind of microscope. And if you see LM, that means light microscope. So this image was probably done on a light microscope. Because we would have looked at cells like this under the scope in lab. And these are cheeks. Well, these are actually cervix cells, but your cheek cells, if you take a little bit of swab from the inside of your cheek, they look exactly the same. So these are what we call epithelial cells, and they've been stained with something called methylene blue, because your cells are not normally blue, but the methylene blue stains darker in the nucleus, so that makes it useful. On the left, you see normal cells, I don't know, 6 or 7. They're a little crumpled up. And on the right, you see these cells have human papillomavirus, so you can see that they're very abnormal. So this is an example of cells obtained from a pap smear. So that's what the pathologist is looking for, abnormalities in the epithelial cells, and just by using a light microscope you can tell if there's any abnormalities. All right, so when you look at this image, what they have is they show the range at which we would usually use certain types of microscopes. So you would use some type of electron microscope for the range of items that's very small. So really structures within a cell, all the way down to some of your macromolecules can be imaged with an electron microscope. Also virus, which are much smaller than cells. So here's flu virus here. You can see bacteria well with an electron microscope, but there's an overlap. So light microscope, that's the kind that we would have used in lab. And you can see individual bacteria, but you can't see detail within a bacterial cell using a light microscope. But you can see them. They're pretty small. You can see some of the larger cell structures, like the nucleus, for example, on that last slide. You can see whole cells. And then with the naked eye, I think you guys know, there's not very many eggs. Eggs actually are fairly large cells. There's a few egg types that you can see possibly with the human eye, frog egg or a fish egg being fairly large. But the naked eye is very limited. All right. So you have to use an electron microscope if you want to see tiny, tiny things like a virus or the parts within a bacterial cell or within a eukaryotic cell. Okay, there are about 20, maybe, it's hard to guess, but 20 to 30 trillion cells in the human body. And there's so many cells, everything has to be managed. Each cell has to have nutrients delivered to it. Each cell has to have wastes picked up. Each cell has to have oxygen provided. So there's a lot of cells. It seems like it would be more efficient if we had less cells, but maybe they could just, each of those cells could just be a little bit bigger, and then there would be less separate cells to manage. But it turns out that there's a rule, of course. There's always a rule. And the rule for why cells have to be the small size that they are is that there must be enough surface area to provide enough points of entry for the oxygen and for the nutrients. And there must be enough surface area to provide points of exit for the wastes. Okay? And so it turns out if you kind of take a theoretical cell, this is just conceptual, if you had a cell, for example, that was 1 millimeter, like basically what they did was they took a cell and squashed it into a cube shape, because that makes the calculation of surface area and volume much easier. So if we took a cell and squashed it into a cube shape and it was 1 millimeter on each side, then you'll remember your surface area of a cube is 6 times the surface area for any one side, which is length times width, right, for each side. And if length and width are 1, then you get a surface area of 6, all right? So surface area of what we're calling the small cell is 6. Now this one, surface area of the larger cell, imagine a larger cell that's 2 by 2 by 2, then each face would be 4 millimeters squared times 6 is 24. Surface area is 24. So the larger cell does have more surface area, but when you divide by the total volume of the cell, you see what we call the surface area to volume ratio. So the surface area of the smaller cell is 6, the surface area of the larger cell is 24. But when you divide by the volume, remember volume is length times width times depth, so 1 times 1 times 1. So the volume of the small cell is 1, but the volume of the larger cell, 2 times 2 times 2, is going to be 8. So the surface area to volume ratio of the small cell is 6. The surface area to volume ratio of the larger cell is actually only 3. What that means is there's not as much surface area to service all the volume of the cell than in the large cell compared to the small cell. The small cell has much more surface area for each unit of volume. Another way to think about this is, let's say you have a piece of, let's say a thick piece of meat, like chicken, and I apologize to the vegetarians in the group, and you may not be able to relate to this, but let's say you have a really thick piece of meat and you want to cook it, and you want it to get cooked all the way to the middle, but if it's really thick, you've probably done this before, you put it on the grill or you put it on a pan or you put it in the oven, and the outside gets cooked right away because the heat is is penetrating just the outer surface, but it takes forever for the heat to get all the way to the middle. In fact, the outside is burnt by the time the inside gets cooked. And that's because it takes a long time for the heat to move all the way to the center. But if you cut that big chunk of meat into smaller cubes and put those in a pan, each one of them has more surface area, and the heat can get into the center of those smaller cubes quicker and without burning the outside. And so in the same way, if a cell is really large, then you can't get the oxygen all the way to the center very quickly. But if instead of having one large cell, you have more cells, but they're smaller, each cell is able to get their oxygen and their nutrients to the center much more quickly. And as you remember when we talked about enzymes, there's a speed at which life needs to move. So the oxygen needs to be able to get to the center quickly. There's not a lot of time to wait. If the cell is too large and the oxygen's not getting all the way to the middle because it's a long distance, then cells start to die. So having smaller cells, the cells that we have are the right size through billions of years of evolution to allow that efficient delivery of oxygen and the efficient delivery of nutrients all the way to the center, from the surface all the way to the center of each cell, and it allows that to happen fast enough for life to continue. So but we say that it's the surface area to volume ratio. The larger surface area to volume ratio, the better. All right, so let's talk about bacterial cells. Bacterial cells are pretty straightforward in a general sense. Now there's tons of different types, species of bacteria, and a lot of them do some really cool different things, but in general we're looking at the fundamental structure of a bacterial cell. What you want to think about is inside the cell, all cells have a plasma membrane. Oops, just this part here. And within the plasma membrane there's cytosol, which is the liquid part. And it's mainly water with some proteins and things suspended in it, dissolved in it. Then you have the ribosomes, which you remember, those are the structures that build proteins, that make peptide bonds between amino acids. And then you have a big, fairly large chromosome made of DNA, and it's just all bundled up. We usually call the place where the chromosome is found in the cell the nucleoid. But it's not a separated part of the cell because bacterial cells don't have any membranes other than the plasma membrane. So it's sort of like if you have some yarn and you kind of twist it up and put it in one spot, it's there, it's still interacting with the rest of the fluid inside the cell. We call that the nucleoid region. That's where the DNA is. And then just outside of the cell membrane, we have a cell wall. Now the cell wall is not made of cellulose or chitin, because remember, cellulose makes plant cell walls and chitin makes fungus cell walls, but those are carbohydrates, and the cell wall of bacteria is also a carbohydrate. It's called peptidoglycan. Well, don't write that down. But anyway, the cell wall is made of a carbohydrate, but it's not any of the carbohydrates we talked about in chapter 3. So anyway, but it's a rigid structure that holds the cell, supports the cell. All right. And then there's some other structures that cells sometimes have, something called the capsule, the pili, flagella for cells that are motile. But those are, like I said, some cells have those, some cells don't, in terms of bacteria. But bacterial cells always have a chromosome bundled up in the area that's called the nucleoid, the ribosomes, which make proteins, the liquid portion of the cell, which is cytosol, a cell membrane, a plasma membrane, and a cell wall. So those things are always there. So I'm putting boxes around the things that they always have. If there's a capsule, if there are pili, if there is a flagellum, some cells actually have many flagella. So the plural of flagellum is flagella. And But that's a focus for your microbiology class. All the different types of bacteria. It gets interesting, but it gets complicated too. Now, what about the eukaryotes? In this course, we're going to talk about plants and animals. Of course, you have the fungi, but we don't talk much about fungi. And we will talk a little bit, even in this chapter, a little bit about the protists. But in terms of like being able to identify structures from a diagram, you're going to have to know the plant and the animal. So this is in your book, but this is the animal cell here. And this is the plant cell here. All right. So in terms of structures, just the ones that I go over in the next slides of this in this lecture would be what you would need to know for the exam. I'm going to leave a few of them off. I'm only going to mention mitochondria briefly in this chapter, and I'm only going to mention chloroplasts briefly because we're going to talk a whole bunch about them in later chapters. And I'm going to hit on most of these things that are here. And so as I go through them, I'll give you some practice pictures that you can practice labeling. So it's very similar to these pictures here. But we're not going to have to label the parts of a fungus cell, and we're not going to have to label the parts of a protist. But they're eukaryotes as well, so they're not hugely different, other than a few places. All right, structure of the plasma membrane. The plasma membrane, we're going to talk about what's called the fluid mosaic model in Chapter 5. So by the time you get through Chapter 5, you will know all the structures of the plasma membrane. But all cells have a plasma membrane. It is the thing that holds what's inside the cell in and what's outside the cell out. So a cell might have more sugar inside of it than outside. Or, and so, but it's not a closed system. So things are able to get across the plasma membrane, but there are restrictions. If the plasma membrane was a closed barrier, then the inside of the cell would be a closed system. And remember, that doesn't exist in biology. So it's an open system in the sense that it is a barrier, but it's not completely shut off. There are these channels, which you can see here. You see these little tube-like structures. These are made of protein, and they create little pores that pass through, like a tunnel that goes through a mountain. When you're driving, you can go from the inside of the cell out or from the outside of the cell in through these protein channels. So those structures are there, and then some other things. But like I said, we'll get into the detail of the structure of the plasma membrane in Chapter 5. All right, let's talk about the nucleus. And really, I've already gone over the very few kind of standard items that all bacterial cells have. And so from here on out, we're looking at eukaryotic cells. As you'll remember, there are the prokaryotes, which includes domain bacteria and domain archaea. And then there's the eukaryotes, which includes domain eukarya. And domain eukarya includes animals, plants, fungus, protists. There's some other things too even, but those are the big ones, the big groups. So in the nucleus, and usually the nucleus is pretty much in the middle of the cell, although there's some exceptions to that, but let's think about it being kind of in the middle of the cell. And the nucleus of the cell is a membrane-bound organelle. That means it's a structure surrounded by membranes or separated from the rest of the cell by membrane. And it's the same kind of membrane as the plasma membrane. It's a phospholipid bilayer. It has pore proteins in it. And so those pore proteins, those channel proteins, are very restrictive in what they let into the nucleus. So transport into and out of the nucleus is heavily guarded, but it's possible. So in this image, you see the nucleus is shown, I guess, in purple. And there's always a dark spot inside every nucleus, sometimes more than one. And the dark spot is called the nucleolus. Now, some people pronounce it nucleolus. Same thing, nucleolus. The nucleolus is where the rRNA is being made. So rRNA is used to make ribosomes. So part of the ribosome is being made inside the nucleus, which is kind of weird because the ribosomes themselves, once they get put together, are outside of the nucleus. But parts of the ribosome are built inside the nucleus in what's called the nucleolus. The DNA in the nucleus is wrapped around proteins, and that material is typically called chromatin, although there's not a huge difference between the word chromatin and the word chromosome in the sense of what it is. Chromatin is just the material. A chromosome refers to a specific, like one DNA molecule is a chromosome. One DNA molecule wrapped around tons and tons of protein makes up a chromosome, but we call the material in general chromatin. The liquid part of the nucleus is called the nucleoplasm. So you have the nucleoplasm, you have the chromatin, the chromosomes made up of chromatin, I guess you could say, and then you have the nucleolus, the darker spot where the rRNA is being made. Now, guys, there's enzymes in here because remember, if you're building anything or breaking down anything, enzymes are involved. So you've got all the enzymes and things that are needed for that in the nucleus. That kind of goes without saying. Now you have these, you not only have one membrane surrounding the nucleus, you have two. So it is a double membrane. It's a membrane, if you imagine like a sphere, and then a larger concentric sphere surrounding that. So it's a double membrane. And there are pores that go through both membranes, and they're called nuclear pores. And nuclear pores can open and close. So they don't just let everything in. They can close and they can open depending on what's coming or going. So there's a lot of research trying to figure out how does the pore know when to open or when not to open. We don't know the answer to that completely. So the double membrane that surrounds the nucleus, you can call it the nuclear membrane. Sometimes it's usually referred to as the nuclear envelope. So I don't know why there's sometimes multiple names for structures. So the nuclear membrane or the nuclear envelope. All right. So that's the nucleus. Now the outer membrane of the nucleus, which is kind of shown here in more of a blue color, is directly connected. It does make the outer membrane of the nuclear envelope, and then it sort of continues and flaps outward to make other parts of the cell called the endoplasmic reticulum. And it flaps outward and makes these little compartments that are shaded in blue. On the outer surface of the compartments are ribosomes, shown in red. And the space that's completely surrounded by the membrane of the endoplasmic reticulum is shown in blue. And it's kind of this continuous space that moves, and actually this structure moves or spreads, goes throughout the cell. The endoplasmic reticulum. Before I talk about the endoplasmic reticulum, I want to go back and talk a little bit about chromosome structure. So if you have a chromosome, which is shown here, if you sort of unravel it, if you kind of pull on the end of the DNA and unravel it, what you would find is the DNA is wrapped around proteins called histone proteins. And so a certain amount of DNA wrapped around histone proteins, the material itself, any amount of the material is called chromatin, but a certain length of DNA makes up one chromosome. In humans, the chromosomes are all different lengths. So you got, the longest chromosome in the human genome is called chromosome 1. but you got one of your chromosome 1s from the egg. Remember when you were an egg and a sperm? And then both parts of you came together. So the egg had a copy of chromosome 1 and a copy of chromosome 2 and a copy of chromosome 3, etc. They've colorized these artificially. They're not really these colors. So there's 23 different kinds of chromosomes that came in the egg. There's 23 that came in the sperm. And so when sperm and egg fuse together, we call that fertilization. And you have 2 copies. Every nucleus of your body contains 2 copies of chromosome 1, one from your mom and one from your dad, 2 copies of chromosome 2, 2 copies of chromosome 3, etc., all the way through 2 copies of chromosome 22. And then the 23rd pair of chromosomes is either an X and a Y or an X and an X. I kind of wish they hadn't named the sex chromosomes X and Y, because all the chromosomes kind of have a shape of an X. Even chromosome 1, even chromosome 2. I wish they'd called them something else, like chromosome, I don't know. Yeah, not M and F, that doesn't work either. Yeah, anyway, they didn't ask me. Okay, ribosomes. Ribosomes make proteins. So one thing I want you to connect right now is the idea that ribosome contains within it the enzyme that does the dehydration synthesis for DS, dehydration synthesis, that makes the protein. So you have amino acids, you guys remember, the amino group of one amino acid and the carboxyl group of the next amino acid interact to form a water molecule, and that connects those 2 amino acids with a peptide bond. And that happens again and again and again, so that in the end you end up with this long chain of amino acids. And so the enzyme for that particular dehydration synthesis is part of the ribosome. And the ribosome is the only way, ribosomes perform the dehydration synthesis reactions. This is the only way cells can make proteins. So if we're talking about a cell making proteins, we're talking about the ribosome doing dehydration synthesis. Now, where do the amino acids come from? Well, they're in the cytosol. They're part of those. Remember, I said there's nutrients and molecules in the cytosol. So they're there. All right, I told you I was going to briefly mention something about the mitochondrion. We're going to spend a lot of time talking about the mitochondrion, so for now, all you need to know is that the mitochondrion is involved with, it's a structure that has a double membrane, kind of like the nucleus. Double membrane is what I'm trying to write. Well, that's terrible. And it's involved in something called cellular respiration. Cellular respiration. Respiration. Now, I know you've learned that the mitochondrion is the powerhouse of the cell, but you know, that's a good kind of 6th grade definition, but we're gonna take that much further. So chloroplast, again, we're going to spend a whole chapter on chloroplast, so I don't need you to know the detailed structure, but a chloroplast is also a double membrane. It has a double membrane around it, and this is where photosynthesis takes place. Photosynthesis, PS. That's all you need to know. You don't need to know the structure right now. We will get there. Okay, so let's go back talking about our nucleus. Remember our nucleus? Remember it was purple? I'm glad they kind of were consistent here with the color. So the nucleus contains chromosomes, contains the nucleolus, which is really just a dark region, a really kind of area of really compacted material where the RNA is made. but and the outer membrane is continuous with the ER. There's two types of ER. There's rough ER, and by the way, ER means endoplasmic reticulum, and there's something called smooth ER. In this picture, we're looking at the rough. The smooth, it's connected to the rough, but it's cut off in this picture. In any case, all of the membranes shown in this image are either directly connected to each other or indirectly connected. So you have the nucleus, the nuclear membrane, and specifically the outer membrane is directly connected to the ER membrane. And the ER membrane is indirectly connected to the Golgi. And the Golgi looks like this. It looks like a stack of pita bread. And then in this image, you're seeing sort of a slice through, like the way it would look in a transmission electron micrograph, like a slice through. In real life, it's 3 dimensions and you can't see the inside of these compartments, but in this concept, you can see inside. But it looks like a stack of pita breads, and there's a little vesicle, a little circle of membrane that will bud, kind of push off of the rough ER and surround a little bit of material from the rough ER, and then move to the closest section of the Golgi, which is called the cis face, and it will fuse, the membrane will fuse like 2 soap bubbles that fuse together to make one bigger soap bubble. The vesicle will fuse to the membrane of the Golgi, and that will dump the contents of the vesicle into the lumen of the Golgi. Lumen means the empty space inside. L-U-M-E-N. And what happens in the Golgi is whatever is in the vesicle enters the first compartment of the Golgi. And what happens in the Golgi is we call sorting and modification. It's amazing, actually. The Golgi, all this different junk comes in from the ER. All these different vesicles are fusing to the Golgi from the ER, carrying all different stuff. Dumps it into the first compartment of the Golgi. Somehow in the Golgi, and there's a lot of research that's they know quite a bit about this, but it gets very technical. But inside the Golgi, the different molecules get sorted and moved around like in little piles, kind of like at the post office. Everybody dumps their letters in the mailbox, and then in the post office they have to make piles. Like this pile is going to, the New England, and this pile is going to the West Coast. So everything gets sorted so that then those little piles move by vesicle to the next compartment. And then in the next compartment, they get modified. So there's sorting into piles and then modified. So things that need to go kind of into the same eventual location get sorted together. And then they get modified. And modified means trimming something off or adding something on. Trimming or adding. So chemical, like little functional groups. You can think of functional groups. Trimming off things, adding on some other things. So it's weird because all this different stuff comes in. Somehow the Golgi knows where each of these things needs to go. It sends it to the correct compartment. And it can go to any of these other compartments. And when it's done, somehow the Golgi knows when it's done, and it gets sent off by another vesicle that buds off of the Golgi. And in this image, they're showing it going to the plasma membrane. It doesn't have to be that, but plasma membrane is one of the final destinations. So another destination is there are some vesicles that bud off of the Golgi, and then they stay in the cytosol of the cell, and they contain enzymes for hydrolysis. Remember, hydrolysis is how macromolecules are broken down. So these little vesicles full of enzymes that are needed for hydrolysis are just kind of floating in the cytosol, and those vesicles are called lysosomes. L-Y-S-O, lysosomes, S-O-M-E, lysosome. Okay. So some of the vesicles that are full of hydrolytic enzymes stay in the cell as lysosomes. Some vesicles will go and fuse to the plasma membrane to release the contents of the vesicle out. Because there are some things that the cell makes that are made inside the cell but need to be released, such as insulin. Insulin is a protein that's made inside a cell, in your pancreas cells. It's made, and it goes from it's made in the lumen of the rough ER, then it goes by vesicle to the Golgi, and it gets modified and sorted, and then eventually it gets released from the cell. And so insulin is made inside the cell, but it needs to be released into the blood, because you guys probably know it helps control your blood sugar levels. So insulin is released through this process. All of these membranes, because they're connected directly or indirectly by vesicle, the whole group of these membranes is called the endomembrane system. The process that I've described to you of a product like insulin moving from the lumen of the rough ER by vesicle into the Golgi and moving into different compartments of the Golgi and eventually being released from the cell, that process is called secretion. Secretion. And you may remember from chapter 6, secretion means the release of something good from a cell, useful. And this little end part is it's called exocytosis, but specifically secretion. So we've got a lot of stuff going on here. We've got the concept of all these connected, directly and indirectly connected membranes being called the endomembrane system. Now notice what's not in the endomembrane system. Mitochondria are not part of the endomembrane system. In a plant cell, chloroplasts are not part of the endomembrane system. So there are some structures, some membrane-bound organelles that are not directly or indirectly connected to this system, which is kind of interesting. But this group of structures are all part of the endomembrane system, including the lysosome. All right. Okay, the Golgi, I think I've already explained what the Golgi does. This is a transmission electron micrograph, TEM. So it gives you an impression that it's a slice through a cell. It's black and white, but this is an actual micrograph from the Golgi of a real cell. So you can see the vesicles coming in to one side and coming out of the other side. But really what they're doing is fusing, and then a new one buds off. But Golgi is where proteins or other cellular products are sorted and modified and then sent out. Sometimes we call the Golgi the post office of the cell. Okay. But hopefully the U.S. post office doesn't, it does definitely sort your mail, but I hope it doesn't modify your mail before it sends it out. That would mean it would open your letters and change things, because in the Golgi that happens. So the analogy works pretty well, but not perfect. All right, so let's talk about a process called intracellular digestion. Intracellular digestion. Intra means within the cell. There is something called extracellular digestion outside of a cell. So this is digestion inside the cell. Now guys, digestion is the same as hydrolysis, the hydrolytic breakdown of macromolecules. hydrolysis. So you still have to know what hydrolysis is. The breakdown of macromolecules into monomer components. So intracellular digestion just describes how does hydrolysis happen inside of a cell. Because the cell itself is made of proteins and nucleic acids and carbohydrates, so it's not like the enzymes for hydrolysis can just be allowed to roam free inside of the cell, because then the cell would digest itself. It would start breaking itself down. So the hydrolytic enzymes have to be contained. And like I mentioned before, they're contained in a membrane, in a vesicle, or in an organelle we call the lysosome. So the lysosome is just a membrane-bound vesicle, just a vesicle full of hydrolytic enzymes. So enzymes that break down protein, enzymes that break down nucleic acid, enzymes that break down carbohydrate. Lipids are actually typically end up in the peroxisome for breakdown, but we're not going to worry too much about that at this point. So let's say the cell takes in some food, some macromolecules. We're just going to call it food. And that process is called phagocytosis. So taking in food into a vesicle, and we usually call it the food vacuole, not the food vesicle, although I couldn't care less. If you call it the food vesicle, it's fine. But for whatever reason, it's become standard to call it the food vacuole. So the food vacuole is the vesicle that comes into the cell by endocytosis, phagocytosis, that brings the food in. Then you have the lysosome. Remember I said it was just kind of hanging out in the cytosol. And then when that food vacuole comes in, somehow it gets stimulated to move towards the food vacuole, or maybe the food vacuole moves towards it. But in any case, they end up fusing together. So you have these two vesicles fusing together, and functionally what that does is it mixes the hydrolytic enzymes mix with the macromolecules so that all those hydrolyses can happen. And the coolest thing is when the two vesicles fuse, if you get a picture under a microscope, like a micrograph of this at that moment, I'm not kidding, it looks like a hurricane. You know how a hurricane looks when you see the picture from space, like this huge storm? I'm not kidding. If you look at the way a lysosome, after it fuses to a food vacuole, the way it looks is like there's a hurricane happening in there. And I think there is. I mean, I think there's just massive amounts of breakdowns happening, just destruction, just on a microscopic level. Really small, but destruction. I mean, that's what hydrolysis is, isn't it? But in any case, anything that is, any of the monomers that the cell can use will actually then be passed out of this structure into the cytosol. So we typically call the lysosome by itself the primary lysosome. Primary. Gosh, I'm having trouble with this. Primary. And then once it fuses to the food vacuole, that larger compartment where the hurricane is happening, that's called the secondary lysosome. Secondary lysosome. Secondary lysosome. So a primary lysosome, there's no action. All the action is in the secondary lysosome. And that's what you get when it fuses with, I can't write with my mouse. But anyway, after the monomers are produced from the macromolecules, then the monomers are allowed to leak, L-E-A-K, across the membrane into the cytosol. So the secondary lysosome allows the monomer units to leak out. And then whatever's left inside the secondary lysosome is waste, undigestible material. So for the cell, it's waste. Just like for you, anything that comes out, that's waste. So that's stuff that could not be digested. And so in the same way, the cell needs to release that waste, and so it releases it by a process called excretion. It's the release of the waste. It is another way, it's another kind of exocytosis. We talked about secretion of insulin. Now we're talking about excretion of waste. So still a fusion of a vesicle to the plasma membrane to release the waste, excretion. So secretion is when a cell releases something by exocytosis, but it's something good and useful like insulin. The cell made it, and then it needs to be released so the body can have it somewhere, so it can move somewhere else. Excretion is waste. It's typically connected to release of waste. All right, I'm going to take a break, and we'll pick up the next couple slides on another section. It'll be pretty short though.